Alright, I'm Trevor Beckett,
I'm Stephen Moldonado.
and this is our project using Python programming to convert RNA codons into their amino acid counterparts and vice versa. Oh, and our mentor is Dr. James Anderson at the moment. Like next week might not be, we don't know yet. And sorry if you're watching this and you know, and you still are, sorry. Okay, so, an introduction to RNA codons. Basically, we're going to make a program that's going to do this right here. It's just going to make it more efficient so that you don't have to look at that table and you can just type something in. Like you can type either this in and it's going to change into this, or you can type this in and it'll change into these, basically. So it's like a more efficient way for researchers to use RNA, or like, you know, in their experiments. And so RNA codons, they make up genetic code of most living organisms, you know. The codons are the three bases, like they're composed of three bases, but these are the four of RNA, uracil, cytosine, adenine, and guanine. And so, you know, as opposed to DNA, which has thymine. And the order of the bases is what determines the amino acid. That's why there's different combinations that make different, you know, acids.
Okay, so our objectives for this is to use Python as our language and to create a system that will convert the RNA codons into the codons corresponding amino acids, and also vice versa as well. And also to make it as easy as possible to use as far as the interface, to make it simple and easily accessible. So, and our hypothesis is to make the program and for it to convert the RNA codon input to its amino acids, and again, vice versa, and to make it all possible with the amount of experience we have with the language itself already.
So the way we're going to go about this, first we're going to research into RNA codons, make sure that we know like what the amino acids do, do some research into stop codons and what we want to do with that within the program. Then we're going to make a plan for how the program is going to work. Make sure that we know which functions we want in there, like the stop codon thing. Make sure that we know just how everything's going to work before we write it. And then using idle, which is what it looks like that over there. And it stands for Integrated Development Environment. Some people also use PyCharm. We're pretty sure we're going to use idle though. We'll write the program. If we have any questions, we'll go to our mentor, of course. And after the program is complete, we'll reread the code. We'll debug it if there's anything wrong with it, and run it to make sure there's nothing wrong. And relevance to biology. Okay, so like I said, the four bases are cytosine, uracil, guanine, and adenine, and RNA. RNA makes up the genetic code of life. Plays a vital role in transcription and translation, which turns DNA into the amino acids that we're using for this, basically. And it carries genetic information around the cell, like with transfer RNA and messenger RNA.
And these are all the institutions that could potentially use our program if it's effective, because pretty much since what we're doing is we're just making it easy to use and easily accessible, it's really easy for these institutions to use them, since we're pretty much just putting a bunch of data and making it easily convertible. So
yeah, and this is just like within Florida. There are like, you know, international organizations that could use it as well, but these are just like smaller laboratories within Florida, so it would be local first. And those are some citations. You know, if you guys really want to know, there they are. And yeah, here I'll go back to the beginning in case people still need that. Any questions?
All right, questions?
Scott?
Has anything like this ever been done before?
I'm sure it has been, because I mean it seems like such a need for laboratories. We haven't like looked into it yet, I guess technically, because because it would just be like a small program within a laboratory to find. So I'm sure there's something like it. Like the table is basically what we're doing, and it was basically made as an easier way to do it. So we're kind of trying to make the easier way even easier, if that makes sense. But as far as I know, I haven't seen like any specific instances of a program like that, but I'm sure it might be out there.
Yes, have you, in your literatures, I know you have posted some of the literatures that you have looked into. Relative to what he just asked, like the GenBank, for example, I think they have now a lot of sequences, but these are DNA. I know that these are out of the PCR, right? When they do DNA analysis of a genome, for example, of an alga, then they upload that, so it's all in its DNA format. So I don't know if there's anything that is doing what you guys are intending to do. So what are you envisioning? You can use those DNA that's already available in the genome sequence and then use those to do what you guys want to do? Is that the main objective?
Basically, it's like working off the assumption that the researcher already has like
The DNA sequence.
Yeah, the DNA sequence and the like the RNA codons already, but they know what the different
What's the
sequences are.
corresponding codon for each of that?
Bases.
Yes.
Okay, like that they know.
So that's already in place.
Yeah, that they know either this
Yes.
or this, but they need to change it. They need to convert to the other one.
Okay.
And so it's just, instead of them having to go to a chart, they'll just have a program where they can type it in, and then it'll change
okay.
it
So
for them.
make it easier
Yeah.
that way. Okay, and that's the one that's not in place, what you guys wanted to do. Oh, cool. That would be really helpful. So, again, by November, are you gonna come up with something, like an example of like one genome of, one species, or what's the goal? Like in November, what can you come up?
Yeah, I mean,
Hopefully.
we're pretty sure that even in the short time period, we could get a program like made by then. And once we have it made, I mean, we could do it for whatever sequence we wanted. So, I mean, we could, if there's like a specific one that we wanted to do, then I guess we could.
Yeah, I think it seems like it should be kind of easy for you guys to be able to get something once you know the program, right? You still have, how much knowledge do you still really have to go through to be able to make this working? Do you need to have a programming class or something, or
I mean,
you're
No, you
already
just need to
in
have-
there?
You just need to have a certain extent of knowledge on how to use it. You don't
Yeah.
need to take a course.
Right.
You can just learn online.
So it's doable, it's
Oh,
really
yeah,
doable.
of course.
Okay. Oh, so the challenge is, I guess, since I think you can really do this, is to identify what would be an interesting, or genome that you want to start looking at, is it a human genome? Is it, and it's at your discretion now, right? To try to see which one would draw more attention. Like you said, if somebody's gonna use this, are you going to be using something in the human genome, or are you going to be using something that's more local? I think that would be something good to now look into and identify, so that in November you're targeting something.
yeah, I mean, once it's made, I mean, it can just be used for probably whatever.
Right, of your
species
interest
you want,
already.
yeah.
Yeah. I think that's awesome. Any other questions? Inputs?
What are your names again?
Steven, Moldonado.
And Trevor Beckett.
We are just on time. Okay, so yeah, thank you so much, guys. If there are no questions,